And our next segment before we go to lunch break is we're going to just recap a few of the Department of Energy biological and environmental research facilities and resources that are available to the community. We've been talking a lot this week about K-Base, EMSL, JGI, Wonders, all of these sorts of things. And so we are excited to just have a few people come and share real briefly about opportunities for how to get engaged with those again. And I guess, some of the talks, about even Ryan's talk right there at the end was a good example. He mentioned Stephen Hallam, his advisor, who's a longtime EMSL collaborator and user. So it's been great to have people engaging with the various facilities and being able to use them. So I think first up in this segment, we're going to have a presentation on JGI, a brief presentation. You've heard a longer presentation on Monday. If you can remember all the way back to Monday, Reka talked to us about JGI and its capabilities, but we're going to have a brief recap of that by Rex Malmstrom. So Rex, if you want to share your screen, or if you're just talking without slides, whatever you're going to do.
I'd have a couple quick slides.
Great.
You can hear me here. Yeah. So yeah, if you can think back to Monday when you heard about the JGI is one of the BRE user facilities, and it's our mission to try to enable large-scale multi-omics studies of plants, fungi, algae, microbial communities in order to address questions related to bioenergy and biofuel and just sort of environmental challenges, biogeochemical cycling and things like that. So we have a number of different capabilities and expertise at the JGI that people can tap into. We do a lot of sequencing of DNA and RNA, but we also do things like DNA synthesis, where we can synthesize genes and pathways in order to test out their products in the lab. We have a lot of metabolomics capabilities. We mine sequencing databases, and we also have some custom sample preparations. And so the way you access these things is actually through a competitive proposal review process. And this is, you probably heard about the user facilities already, but it's a little bit different than like a typical funding scenario where you write a proposal and if it's accepted, you get funds. With the user facilities, if your proposal is accepted, what you get is our services and access to resources. So, and so that's something that if it's accepted, then we do a lot of work for free. The way that people can access these capabilities is through what we call our user program and the community science program. We have a number of different calls for proposals throughout the year. And so if you want to know more, there's this link up here, and this will be sort of your gateway into understanding how to get a proposal into the JGI. But this is one of the pages I just took a screenshot of, and you can see that we have these different calls. Each of these rows is a different call, and they have at different times. And so we have the large scale CSP call with the ficus, where you can write a single proposal to access both JGI and EMSL at the same time instead of writing two separate proposals. But the one that may be of particular interest for folks in this session is what we call the New Investigator Award. We offer it twice each year. And this is really limited to people who've never worked with the JGI before. They've never been a PI on another project. And so it's the idea to have a little bit smaller sort of pilot scale projects to get people in and start getting data. And then, if it really goes well, it can usually then lead to a much larger proposal down the line through one of the other ones. If you notice here, the submission deadline for the next New Investigator one is September. So there would be time to certainly learn more about it if you were interested in those capabilities. And up here in this corner, I have Tanya Woike's email address. She runs the user programs for JGI. And so feel free to reach out to her or anyone else you actually have. If you know anyone at JGI, you can reach out to us and we'll get you the answers as best we can. I was just going to highlight, just because of the nature of the course that you're doing, two of the capabilities in the world of microbes that you might actually be interested in that are a little bit unusual. And they're focused on studying microbes, the functional work of microbes, rather than just sort of a strict metagenomic sequencing and get in the metagenome. You can actually incorporate these methods with experiments. So one of them we call BONCAT FACS, which is a method for selectively sorting, recovering, and sequencing microbes that were metabolically active in your incubation. And really briefly, the way this goes is you incubate your cells along with this modified amino acid called HPG. It gets incorporated into newly synthesized proteins, and because of a little side chain on the HPG, we can then click on a fluorescent molecule, which will light the cells up if they've been synthesizing protein, and then we can put them on the flow sorter and sort just those cells. That can be done for single cell genomics or a targeted metagenomics type study. So anyway, this is kind of a cool technique that you can use, especially if you're dealing with systems where you think that have reason to believe that many or most of the cells might not really be active at any one particular time, or if you have experiments where you think you can make an amendment and you might be starting from very few cells that are active and you really turn a whole bunch on, or vice versa, where you do something where they're mostly active and you hit them and you want to see who can keep surviving that. So anyway, that's one of the versions of flow sorting cells based on activity. Another method we have is stable isotope probing. It's kind of been around for a little while. The idea meaning you incubate your cells with the isotopically labeled compounds, and that isotope eventually ends up in the DNA or RNA of those microbes, and then can be separated based on a density centrifugation to separate the labeled and unlabeled nucleic acids, at which point you can do sequencing and identify who are the cells, get the genomic information of the cells that were actually consuming that compound. Anyway, it's been around for a little while, but it's a real pain in the neck to do, especially at the scale you'd want to. And we actually have automated ways of doing that. So it can be a useful resource for those who want to do SIP, but just having trouble getting over the hurdle to do it at the scale they want. Okay, that was a quick overview. A lot of genome sequencing, again, genome sequencing, metabolomics, DNA synthesis, some custom preps. If you want to know the latest about the JGI, you can follow us on all the different social media channels. And then we also have these two cool podcasts that have been out right now. You might want to check them out. Here's a short little link up here that's highlighted if you want to listen to something while you're walking the dog in the morning or on your commute. Okay, that's it for me.
Great, thank you very much, Rex. That's awesome. Yeah, I'll give a plug for the podcast, the Genome Insider podcast. There's one on Kelly Wrighton's group from there that's really great.
That's a good one.
So, yeah, great. Thank you very much. And next up, we're going to hear a little bit about Ideas Project from Xingyan Chen. Xingyan is my co-PI on the SFA project at PNNL, and she is also the PNL co-PI on the Ideas Project. You've heard a little bit about Ideas, and I wanted to give her a chance to tell you a little more about what it is and how it might be beneficial to you as a community member. So, Xingyan, if you have a couple minutes and a slide for Ideas.
Okay, so let me share like one slide.
Perfect.
Let's see. Where is my screen? So you will probably see that again in like my other talk after lunch, but I can explain more about Ideas right now. So I can save my time. Let me present it, and it does it come up?
I'm not seeing anything.
Oh, okay. Let's see. Okay, this should be.
There we go. Now I'm seeing it.
Okay. So.
Yeah, there you go.
Yeah, so at the Ideas, it's a short name, represent the interoperable design of software ecosystem. And this project focuses on building such software ecosystems for watershed sciences. And you have heard a lot about like biogeochemistry and how the microbial processes and all those genomic informations are impacting those biogeochemical processes. So as we, I don't know how much watershed models you have run so far, but a lot of times we start with hydrology in watershed hydrology models, watershed models, and then we add like biogeochemistry and other processes like land surface fluxes, evapotranspiration, and all this to our models. And in the past, the community has developed like a lot of different codes, as you can see all those names over here, including OpenFOAM, including pFlowTran, and Wolf Hydro, CLM, SWOT, and all these different names. And those models are usually designed to represent certain processes for the problems that are driven the development of these. And currently, not a single code can do like all the processes we're interested in linking watershed hydrology with the biogeochemistry and make them fully coupled with all the components in the watershed coming into play. So what ideas watershed is trying to do is building like the common libraries underneath all those codes. In that way, we are trying to make like different modules within like a code, like sharing those libraries so that they can be like plug and play ideally, but it's like very difficult to make that just like, it doesn't happen just like a magic, but we need to like build like interfaces to make different modules talk to each other. So with that like common library makes such like integration or building the interface between different codes and processes much easier. And this is like an effort we cannot do without the community involvement. So what we're doing is like we collaborate a lot of developers of all these codes from like different universities, different national labs. And then we design the system to make the interface like as agnostic as possible to talk with a lot of different models that do the similar things. And we also will have like, say, a lot of activities designed to compare the model performances with different configurations, use the same set of the codes, or use like different codes as well. And we run simulations, we compare the simulations with models, simulations with data. All these are integrated to help us build like more accurate predictive models for predicting what is happening within the watersheds and how that is impacting, say, the water quality and the quantity that impact our daily life. So with that, I can take questions if anyone has.
Thank you, Xingyan. I think we'll ask people to take the questions over in the Discord or in the Zoom chat.
Okay.
And, I appreciate that. And please come back for Xingyan's presentation. She'll be the first presenter this afternoon after the lunch break. Our next person up is going to be Charu Varadarajan from Berkeley Lab, and she is going to talk a little bit about ESS Dive. So we've been working this week with Wonders data, which, as you know, we downloaded from ESS Dive. And so Charu is going to join us for a few minutes and talk a little bit about that system. Thanks. Hi, thanks for joining us, Charu.
Thank you, Tim. So I'm really glad everyone's gotten a chance to work on ESS Dive. I'm Charu Varadarajan, and I'm one of the co-PIs for ESS Dive. And sort of just to step back a little bit and tell everyone about what ESS Dive itself is. It's the data repository for this DOE-sponsored program for terrestrial and subsurface ecosystem research. So that's called the Environmental Systems Science Program. And what we store here is a very, very diverse data, everything from genomics, but also, you know, sort of mineralogy, geochemistry, hydrology, so, as well as remote sensing and modeling. So I'll take you through a quick tour of our data portal, but just want to say that this is sort of a very diverse data repository. So you can find not only the genomics data out there, but also sort of related ancillary data that you may want for your analysis. So I wanted to quickly actually just show our website because I think for someone who's trying to use ESS Dive, this is probably one of the best places for you to go to for our resources. So I want to point out that a couple tabs here. One is for users where we have a lot of guidelines for things like how, if you wanted to store with ESS Dive and you're putting in a proposal to DOE, how you would go about doing that, but also citation guidelines when you're using data from ESS Dive and how you would contribute data. We also have extensive support and a help documentation, as well as video tutorials for some of you who want to get a bit more in-depth, deep dive into some of the features that we're offering. So the data portal here would take you to our main data portal here. And so I gather you've already had enough experience playing around and downloading this. But if you just do a quick search on metagenomics, I want to point out that there's a lot of very diverse data here that you can start to download, not just from Wanders, but many other DOE projects that are collecting related data sets. So I'm going to, since you've played around with Wanders, I want to click on something else that they'll show you a little bit about what it has. And also you can see sort of the spatial view. You can do lots of other filters in here. And I think that some of the aspects of, you can find the citation, copy it, put it in your manuscript. You can see how many downloads or views there are. And when these data get cited, the citations will start to show up in their citation count. And we have lots of information that provides you context for what these are. So anything that's mentioned in here is actually searchable. So if you search, for instance, here it's for JGI, you're going to be able to find this data set through our main search portal. And then you can, of course, download it and get other information about where this data is. So I won't go too much into the details about how you do this, since you've already been done with it. I also want to mention something that's not available through our website is some support we have for more of the data contributor side of things. We're actually right now working on some community projects here for developing data standards for a number of different data types. And these are being done by several of the national lab partners that we have, including some people from the Wanders team that you can see here. And what this is when these are still in the process of being developed right now, but when these are complete, these will help ensure that the data within ESS Dive are standardized. So when you download the data, you can start to write automated scripts that pull some of the information out. So this is still work in progress, but we've really made enough strides that we think within the next few months, we're going to have sufficient data standards out there for samples, as well as for soil respiration, hydrology, and leaf physiology. So, and we also are providing support for the International Geosample Numbers, IGSNs, and working with MSIL, NMDC, and KBase and coordinating some of these standards across our facilities. So that's really what I had to say, and I'm happy to take questions at the end.
Okay, great. Thanks, Toru. Thanks very much for joining us. If anybody has questions for Charu, please drop them in the Zoom chat and or over on Discord. And let's see, I think Amy and James, are you guys on?
I'm here. James is here.
James, did you want to say anything about Wonders in this segment? I didn't know if you were prepared to show a slide or not.
So I do not have a slide prepared. Sorry for that. So Amy gave her presentation on Monday. I guess I'll just say that I think, as most people heard, Wonders really is built for and is enabled by the community. It's not for PNNL scientists or anything of that nature. It's really meant to advance with the community and by the community. I think, as Amy mentioned, our sampling campaigns are very much informed by the needs of the community. We do modify the things we're measuring, how we're taking samples based off of community needs. So we're always looking for input in terms of where the community wants to go. We encourage you to reach out to us if you have thoughts along those lines. You can get added to our newsletter distribution to keep up with what's going on. Always very happy to receive emails from anybody wanting to provide suggestions, criticisms, whatever they may be. You can email us at wonders@pnl.gov. I think that email has been shared. Maybe it can be put into, or maybe I can put it into the chat if people don't have it yet. And there's lots of ways to be involved in Wonders. That's, I guess, maybe one of the key things here is, we send out kits to people to collect samples, and so they come back here and data are provided. And so, you can be involved as someone that wants to go in the field and collect a sample that may be useful to you to learn something more about your own particular field system. You might be able to use the associated data to write a manuscript or write a proposal or both. Very much encourage people to do that. Take Wonders, use it, leverage it towards your needs, towards your goals. That's quite important. I really want to encourage people to do that. But in addition, even if maybe you're not a field person, that's totally possible. We're really looking for people that want to use the data in ways that are interesting to them, to ask questions that are interesting to them. We're trying to move towards a publishing model, really a way of doing science, not just publishing, but a way of doing science in which we're doing it more as a community. So I think people are familiar with the standard way of doing science, where there's an individual investigator, they go get their data, they write a paper, maybe with a few colleagues, publish that paper. Increasingly, those data then are made available. That's great. What we're trying to do with Wonders is make the data available ahead of time, before manuscripts are written. And what we want to start to do is to pitch ideas to the community of, say, hey, we have this idea of a science question. There's some data here that we think would be useful to answering this question. Maybe do this by social media, and start to get feedback on that question, the data, the relevance of the two put together, and then actually pursue the analyses as a collective in an open way where the community is actually contributing analyses, not just feedback, but actually doing hands-on work with the data. Community-based interpretation of the associated results in an open, fully open, transparent way. Whoever wants to get engaged is welcome to do that. Writing the manuscript as a community together. So it's not a single investigator doing this, in isolation, kind of doing their thing, but it's a community together doing it. So our colleague Emily Graham recently did something very much like this in more of a conceptual way with disturbance ecology. Maybe some of you are involved in that manuscript. I think it's in review right now, where it was, she calls it her Twitter paper, where she had an idea of thinking about a new way of conceptualizing disturbances. She reached out to the community via social media, had people from all over the world contributing to that manuscript in an open, collaborative way. And so we want to take that to the next level of analysis and interpretation of those analyses together. So if that sounds interesting to anybody, or wants to kind of help develop even that concept of a way of doing science, please reach out to us. You can ping us on Twitter, email us, whatever it may be. But the more people get engaged, the more powerful it will become, and the more it will help advance us collectively. So I think I'll just leave it at that, and look forward to talking to more of you.
Thanks, James. Great. So our final presenter in this segment is going to be Nancy Hess, who's going to talk about the host facility, EMSL. I also wanted to mention before, as Nancy, if you want to start sharing your screen or getting ready, of course, KBase has been a huge component of this whole summer school, and KBase is a very important resource to the DOE and the broader research community. I just wanted to point out to those who may not have seen the message Alicia posted on Discord this morning. She noted that she had opened up the special group for the summer school to the public as of today. So if you want to access those narratives and all of the material that was used by the students in the private sessions, those are available. And I'll post the KBase website into the Zoom chat for you shortly. So Nancy, thank you.
Is it showing presentation mode or showing correctly? Okay. Okay, yeah, so thanks everyone, and thanks Tim. So I started off with this slide on Monday, but just to reorient you, we're located here in Eastern Washington. We're run by the Department of Energy, Office of Science in the Biological and Environmental Research Program. EMSL has like 150 kind of really unique instruments and about 200 scientists that do work in the environmental and biological science areas. We really embrace this integrative experimental modeling and computational approaches to solve scientific challenges in that molecular and up to ecosystem scale. And we have users from all over the world. And we have a pretty neat website. You can check it out there. Just wanted to talk a little bit more about the type of science that we do. We have a science area called functional systems biology, and we're really trying to explore the prediction and control of biology for sustainable energy. That's taking the information that's encoded in DNA, understanding what is actually expressed in terms of the different biomolecules, understanding how those biomolecules interact in metabolic pathways, to do things like store lipids in yeast cells, Yarrow shown here, or how that's expressed in the different plant phenotypes in terms of bioenergy systems. We're also very interested in understanding these processes and generating exoenzymes that are responsible for the degradation of biomass and understanding how to optimize those processes. On the environmental side, we saw a lot of talks in this area and some great application areas this morning about understanding the interaction of microbes in the soils, how they interact with the rhizosphere and root exudates, and how that can change the soil chemistry and mineralogy, and how that can lead to the buildup and cycling of carbon and other micronutrients in soils. But we're also interested in this amazing connectome between plants and microbes in the subsurface, and how that they're signaling an exchange of nutrients, and tapping into this total information network that goes on there. And also the above ground point. We can observe from space the impacts of water limitation and drought long before there's effects in terms of trees dying. We can see phosphorus limitation in the rainforest using these same mechanisms. So essentially we're trying to decipher these molecular signaling pathways to really understand and predict the ecosystem response to perturbation. And so in EMSL, we have these two areas that we solicit calls for proposals in them, and we support our research by using these integrated research platforms that are called out here. And so the 150 instruments that I talked about earlier are actually assembled into research platforms to do functional omics, to look at bioimaging on a molecular basis. Where are molecules located in cells? What is their transit across cell membranes? Understanding cellular dynamics, the accumulation of lipids in exoenzyme pockets and things like that. We're more in the environmental area. We're looking at those biogeochemical transformations in soils. We have instrument platforms to look at the flux of nutrients and micronutrients using isotopes and different chemical analysis platforms. And we have a molecular phenotyping platform where we have specialized plant growth chambers where we can add C13, we can measure photosynthetic efficiency. We have rhizoboxes and different methods for extracting out pore fluids. And one important part of this that really was highlighted in the school was this computation, analytics, and modeling platform that really wraps all these experimental platforms together. We do have a pretty swift, and we're actually upgrading our supercomputer as we speak. And we're really interested in leveraging that computational strength behind some of our analytical capabilities. So to collaborate with EMSL, we have a couple of proposal opportunities. We have similar to JGI, we have a large-scale research call that comes out in the winter, and that's about a 2-year program. We have the joint call with JGI, the FICUS research program. We've also expanded that recently in a pilot with NSLS2, as well as NSF's National Ecological Observatory network, NEON. So those are opportunities where you can access resources at multiple user facilities and to conduct research. We have an exploratory research program that is year-long in duration. That call should be coming out actually just in a few days, so be sure to look for it on our website. And we also have this limited scope option, which is a great way to do a proof of principle. You just want to see how something works, and you can send us samples. We can work with you to see if you're going to get the type of analytical result that will move your science forward. We're also really interested in developing a more sustained research relationship. If you want to develop unique capabilities in your science and partner with us to co-develop things, we have a scientific partner proposal aspect. So go to our website. You can learn more about these calls in the for user area, and it talks more about these call opportunities. Here's just a snapshot of our annual and special calls. And then I wanted to just end here by mentioning that we have our annual user meeting coming up and in October, and it's going to be visualizing the proteome. We've gotten some really nifty new cryo-EM microscopes and our high-resolution mass spectrometry capabilities to understand the structure and function of proteins and where they're located within cells. And this will also be a virtual workshop. And so with that, I think I'd be happy to respond to any questions that you have over on Discord. So
Thank you very much, Nancy, and thanks again to all the speakers in this segment. We've covered a lot of ground this morning already, and hopefully this has all been helpful and useful to you. There's some information being posted in the Zoom chat about various links and how to connect with the facilities you've just heard about, and some more information over on the Discord site, as well as information in the Google Doc that's been shared this week for other links. So definitely encourage you to contact people from these resources, work with us. One of the questions that came up over in Discord was about, is it possible to work with EMSL scientists before you submit a proposal? And definitely the answer is yes. We encourage that strongly. So if you're interested in an idea, contact us. We can help you figure out which instruments might be useful, or what, how even help you design your experiments and prepare your samples. So